Hello, everyone, and welcome to the Hereditary Disease Foundation's second Research Spotlight webinar of 2022. I'm Megan Donaldson, CEO of the HDF, and I'm pleased to note that we again have a diverse and international group of HD families, advocacy groups, and scientific and clinical professionals joining us today. Most of you are familiar with us already, but for those of you joining us for the first time, the Hereditary Disease Foundation funds research to identify treatments and cures for Huntington's disease and other brain disorders. Today, we are thrilled to have with us Dr. Ali Khoshnan, Senior Research Scientist at California Institute of Technology, discussing our gut microbiome, a topic that many of you may already be familiar with as it relates to our general health. Ali will discuss how to better understand what kinds of microorganisms live in the gut and how they influence Huntington's disease. The Hereditary Disease Foundation has funded Ali's research for the past 2 years, and by joining us here today and helping to support us financially, all of you are contributing to the success of our research efforts. Please feel free to type your questions into the Q&A box at the bottom right of your screen throughout Ali's presentation. He will be available to answer your questions at the end of his talk. It is now my pleasure to introduce Dr. Ali Khoshnan giving a presentation entitled Gut Bacteria and HD, What's the Connection? Welcome, Ali, and thank you for joining us today.
Thank you for the kind introduction, Megan, and thanks to all the attendees for being here. I hope to be able to convince you why we should love our gut. It's pretty hard to believe like some germs in the gut could have such a powerful effect on brain chemistry, brain's function, and our behavior. But the accumulating studies in the last decade or so support the notion that there is a strong connection between the gut and brain, and perhaps in the future it may be possible to identify gut-based therapeutics for brain disorders, including Huntington disease. Let's begin with some terminology. What is a gut microbiome or microbiota? It's simply the collection of microorganisms living in the gut, and that's an ecosystem consisting of fungi, bacteria, viruses, and archaebacteria. However, bacteria are the most dominant species, and usually this is also referred as microbiota, and that's what we will use today. There are over 1,000 to 4,000 different species of bacteria in the gut. You could think of them in terms of mini factories producing health-promoting compounds, obviously when they are happy. That amounts to about 100 trillion of bacterial cells in the gut, which weighs about 3 pounds, equivalent to the size of our brain. And some argue that, in fact, the gut microbes may have a conscience of their own. The gut bacteria carry about 3 million genes. This is about 100 times more than what is in the human genome. So from a gene dosage point of view, we are 99% bacteria and 1% human. What's interesting about gut microbiota that it is plastic, and we can change its composition and chemistry to benefit us. More importantly, we cannot live without these critters in our gut, so they are very, very important to our survival. How do we get gut microbiome? It simply assembles in a ordered and systemic way as we age, and they colonize different parts of the gut. The source is usually the environment, and colonization begins during birth and matures, and the microbiota matures in the adulthood. Young adults usually have the most diverse microbial species in the gut, and it's presumed that diversity coincides with a healthy gut. What factors regulate the composition of gut microbiota? It's usually genes, the physiology of the gut, for example, the nutrient oxygen level, acidity, etc. And obviously the environment also plays an important role, including our state of mind and diet. The bacteria in the gut, they're not just floating in there by themselves. They have practically what is called a niche, or we could see it as a zip code, that they colonize in a specific area of the gut based on their nutritional need and where they could thrive the best and function the best. However, the majority of gut bacteria are in the colon. In the context of a healthy or optimal gut microbiota, remember, one ecosystem doesn't fit all, so we're all practically different in the context of gut microbiota, and likely it is tailored by the human activity. What's important, however, the activity and the composition of the gut microbiota must be balanced. What happens if the balance is changed? A state what is called dysbiosis happens. Compounds that promote dysbiosis include antibiotics, medications, infections, poor diet. So simply what happens in a dysbiosis, the number of friendly bacteria is decreased and the number of hostile bacteria increased. So the ratio of this hostile to friendly bacteria determines the outcome of our gut microbiome. But in general, gut dysbiosis is usually linked to most human disorders. For example, looking in the context of brain disorders, early in development, gut dysbiosis could lead to disorders such as autism, whereas in the adult, it could lead to neurodegeneration and perhaps poor cognition. The beauty of our gut microbiome is that we have control over it. Two elements that modulate the activity of gut microbiome are diet and lifestyle. For example, research suggests that adhering to a Mediterranean diet or a high fiber diet usually associated with the growth of health-promoting bacteria in the gut, and this is linked to better brain health, lower inflammation, better cognition and memory. And in the context of Alzheimer's disease patient, there appears to be less buildup of these toxic proteins or toxic protein clumps. The other factor here is the physical and mental activity. Good sleep and intermittent fasting also correlates with the growth of health-promoting bacteria. On the other side of the cone is the Western or industrialized diet. Adhering to this type of diet is linked to the growth of inflammatory bacteria, which may lead to neuroinflammation, poor cognition, and other biological defects. What other factors that contribute to this kind of what I would call a dysbiotic gut microbiome? It could be stress, anxiety, smoking, and excessive alcohol use. So keeping the gut microbiome healthy is probably one of the best investments that we could do for our health. Well, how do gut bacteria communicate with brain? There are various mechanisms. Here are listed some examples. Some bacteria are able to produce neurotransmitters or stimulate the production of neurotransmitters in the gut. These are regulators of brain activity and control moods such as happiness, pleasure, motivation, and even mobility. Gut bacteria could also produce these regulators of the immune system. In the context of a healthy gut microbiome, these immune regulators are protective. And in the context of a harmful dysbiotic condition, it could again lead to neuroinflammation. Bacteria also produce a large number of compounds that could have a profound effect on gene expression in various organs. For example, BDNF is a neurotrophin that promotes neuronal growth in the brain. It is reduced in HDF, but its levels are modulated by gut bacteria. how all of this information is transmitted to brain. There are different pathways, and it's not important for us to know that, but there are usually direct pathways, that mean direct connection between the gut and brain by neurons, and also it could be modulated remotely by these soluble factors that I mentioned. There is evidence that the gut bacteria of HD patients are different from controls. Two studies support these hypotheses. One from Australia indicates that HD patients have a lower number of bacterial species in the gut, predominantly in males, and that coincides with cognitive decline. Another study from China actually indicates the opposite, that the higher number of bacterial species are present when compared to healthy controls in HD patients. And on the other hand, presence or absence of specific bacteria are linked to inflammation and elevated inflammatory biomarkers. We did a pilot study with some patients from Santiago, Chile, in collaboration with Dr. Chana from Enroll HD. Interestingly, we found some results that they were very similar to what was done in Australian group. We saw low number of bacterial species in these patients, but we couldn't find any overlaps between those differences. So this is just to show how this could complicate, the studying of the gut microbiome in diseased individual. But they are very, very important in a sense that they highlight gut dysbiosis is indeed occurring in patient. But what might be interesting is to find out what is the mechanism, how does gut dysbiosis may occur in Huntington disease. For that, we focused on the function of mutated Huntington. You're probably aware that mutated Huntington forms aggregates and leads to the loss of neurons in the brain. We ask exactly the same question: is mutated Huntington toxic in the gut cells? And for that, we are focusing on two different cell lineages. One is the cell lining of the gut lumen, and the other one, the nerve cells in the gut that they are surrounding the gut. To answer some of these questions, we are focusing on two models. We're using the mouse model just simply because it has a complex microbiota and gut anatomy that is closer to humans. And we also are using the Drosophila, which is cheap, faster, and it's genetically versatile, so we are able to study mechanistic pathways of gut brains in HD. Before I get into what we're doing with these models, let me tell you a bit about the structure and function of mammalian gut. You are probably aware that the human gut is around 16 to 20 feet long. And when we look inside the gut of human, we probably see in the lumen a lining such as this that is covered by this hair-like structure, which are practically folded intestinal epithelium. These folded or hair-like structures are called villi. Let's look at the anatomy of a villus. So what you find, a structured and highly organized arrangement of various cells spatially in the villus that they serve different functions. If you focus on the top, you see the differentiated cells that they are responsible for nutrient absorption, communication with the immune cells and nervous system, and also produce these antimicrobial compounds that could defend against harmful bacteria. These layers also form a barrier against leakage of some of the gut content into other organs. What's the focus of our attention is here in the crypt area of the villus. The crypt has a population of cells that they have stem cell properties and which produce the cells in the villus. You may be aware again that the gut epithelium regenerates itself every 5 to 7 days, and these cells are responsible for the generation of the gut epithelium. Underneath of the villi is the enteric nervous system. This is a constellation of around 200 to 600 million neurons. They are different types and serve different function, but the overall goal of enteric nervous system is to maintain gut homeostasis and practically regulate the differentiation of these cells and the response of these cells to the gut content. Here is some basic preliminary studies. We examined the gut tissue of HD animals for the presence of mutated Huntington. We practically asked, where is Huntington in the gut epithelium? What we found, very interestingly, was that Huntington predominantly is concentrated in these crypt cells in the stem cell region that is responsible for the regeneration of the gut epithelium. And that has led to asking several interesting questions which are in progress. So what we're asking is, what is the mutated Huntington doing to the biology of gut stem cells at a cellular and molecular level? How the level and activity of mutant Huntington is altered by the presence of, let's say, hostile or healthy gut bacteria. What we're asking is practically whether mutated Huntington generates a defective gut epithelium, which could explain some of the problems in HD patients, for example, poor nutrient absorption, gut dysbiosis, and perhaps the presence of a leaky gut, which may allow the toxins to enter circulation, cause damage, and probably induce a systemic inflammation. And obviously, a defective gut may lead to poor regulation of the immune system. We're also looking at the enteric nervous system. You may be aware that mutated Huntington forms these clumps in neurons in the central nervous system, identified here by these red dots. We looked at the enteric nervous system of HD mice, and again, what surprisingly we found that abundance of this clumped protein in the enteric nervous system. The reason we could tell they are there because they co-localize with the markers of enteric neurons, identified here as these blue dots. All of these blue dots here are representative of Huntington clumps in the enteric nervous system. So again, we're asking what kind of neurons are they forming, and are they really causing neurodegeneration, or are they neurotoxic? Are there any connections to toxic protein clumps in the brain? Do hostile bacteria play a role in their buildup? And obviously, could we use either probiotics or prebiotics to prevent the buildup of these compounds? We're also interested in the function of mutant Huntington in the enteric nervous system. You may be aware that mutant Huntington forms clump in neurons of the central nervous system, identified as red dots here. When we looked at the enteric nervous system of HD mice, what we found, abundance of these clumps in the enteric neurons. We knew that they were in the enteric neurons because they co-localize with biomarkers of these neurons here. So the kind of questions that we're asking, what kind of neurons are they forming in? Are they neurotoxics, in other words, killing the neurons? Are there any connection between the toxic protein clumps here and in the brain? Is their level regulated by the presence of absence of hostile or healthy microbes in the gut? And more importantly, can we prevent their buildup by diet or other gut modifying compounds? Let me switch to Drosophila model and show you what we are practically doing in this model. Again, the HD transgenic models, they express mutated Huntington protein, and they simply, in one way or another, they develop HD phenotypes. For example, they form clumps in neurons and cause neurodegeneration. They develop motor defects and become immobile. And obviously, they die faster than the control flies. Some info on the fruit flies' gut. They do have a gut and has some structural and anatomical similarities with the mammalian intestine. They also have an enteric nervous system which connects the brain to the gut of Drosophila, and also have a microbiome, only a handful of bacterial species, however. So the kind of questions are being asked, let's say, how do elevated bacteria, let's say hostile bacteria in the gut affect HD phenotypes, such as protein aggregation, neurotoxicity, and mobility and longevity? Can we reverse HD phenotypes using gut-based compounds, or do hostile bacteria change brain chemistry in HD flies? And practically, what are the circuits for the delivery of this information between the gut and brain? It's important to know that gut flies have very simple macrobiota, and two major families are lactobacilli and acidobacter. It's not important to know the name of these two, but the ratio of these two bacteria is critical for the health of the fly. In particular, elevation of this acidobacter has been linked to inflammation and death. We looked at in the transgenic HD flies for the level of these two families of bacteria. What we found that transgenic HD flies have lower level of lactobacilli and elevated level of acidobacter. So we asked the question, can we manipulate these bacteria and affect HD phenotype? And the first experiment that we did, we put the HD flies on antibiotics to eliminate all bacteria and see what happens to HD pathology. What we found that when flies are on antibiotics, they form less protein clumps in their neurons. We also looked at their mobility, how well do they move around, and for that we used a climbing assay. HD flies have a defect in mobility, and only 25% of them, for example, let's say, make it to the top of this cylinder in a given time. However, those that they have been on antibiotic, they do much better, and they do it faster than those without antibiotic. We then ask what would happen if instead do the reverse experiment. Can we infect these flies with either bacteria that they are the hostile bacteria of the flies, such as acidobacter, or those that they are found in the human gut? E. coli, for example, in the human gut is been associated with inflammatory disorders of the gut and many other disorders. So what we did is we put HD flies on either acidobacter and E. coli and asked the question, what is the outcome for the phenotypes? We found the reverse. We found that those on E. coli had more protein clumps in their neurons. Moreover, they were less mobile. For example, 25% versus 5%. Practically, these flies were really unable to move about after 7 to 15 days on E. coli. This was specific to HD flies. As we put controls on E. coli or acidobacter, we did not find any defect in mobility. What was interesting that E. coli infected flies also die faster. This is a survival curve, and you could appreciate here that those on E. coli, two different strains of E. coli, die faster compared to those where they are not treated with E. coli. So what these data really suggest that inflammatory gut bacteria accelerate HD phenotypes in fruit fly models. However, it is bold really to say that infection could cause HD disease, but it may, if it is real and if we are able, for example, reproduce these in a mouse model, it may be a new way of studying Huntington disease. We then ask, what can we treat these phenotypes with compounds that they have anti-inflammatory activity, but that at the same time have been demonstrated to have gut editing properties. So we found some of those compounds and we fed them to the HD flies and we asked the questions, what happens? When we looked at the protein clumps, we found the flies on therapeutic compounds, they have less of these protein clumps. Their mobility was much enhanced, that they did much better in this climbing assay. And we also found that the HD flies on these compounds, they live much longer. For example, here are HD flies control and those that they have been treated with a compound here, demonstrating that they live longer. What's interesting that some of the compounds also extended the lifespans of those that they were treated with E. coli. If I get your attention here, you could see that the E. coli treated HD flies are like the controls compared to E. coli alone, which die faster. What we found that was interesting, that some of these compounds prevent the colonization of E. coli in the HD flies, which we think might be one of the benefits of these compounds. So this is all nice and dandy, but we really need to get to the bottom of how these compounds, or E. coli, for example, regulate gut-brain interaction in HD. So what we decided to do to look at the genes that are turned on or off in the whole organism. So what we found here, that we found genes implicated in the immune defense and inflammatory pathways that they are turned on. Here is a summary of those studies. For example, both HD flies and HD flies on E. coli had these genes elevated. On the other hand, HD flies that they were fed this anti-inflammatory compound or diet did not show the changes. Why these changes are important? If we look at the function of these compounds, we realize their importance. For example, these compounds are responsible for killing bacteria, regulating immune system, and some of them are even involved in neurodegeneration. On the other hand, we found clusters of genes that they were involved in brain function, activity, and physiology, that they were practically turned off by E. coli predominantly. Here is a summary of those results. Again, HD flies has some reduction in these genes, but when they were infected with E. coli, the fold differences was much different, and E. coli treated flies have much lower levels of these genes. On the other hand, when they were treated with the anti-inflammatory diet, these changes were very similar to controls. Again, these changes were only noted in HD flies, suggesting the susceptibility of HD flies to these manipulations. What do some of those genes do? For example, some of them are involved in neurodevelopment and maturation of the neurons. Some of them control neuronal communication pathways. For example, formation of neuronal contacts between one another, the way that they communicate, for example, in learning and memory or doing a task. Some other cluster of genes were these neurotransmitters, involved in neurotransmitters production and transport. Again, these neurotransmitters are those molecules which activate neurons. One other family of the genes were clustered that they controlled motor behavior. And again, these are very exciting because the genotype, it's overlapping with phenotypes of what we observe in the HD flies. So what do we have? We're seeing that elevation of defense and inflammatory genes in the gut and reduction of genes in the brain. And perhaps these fluctuation in gene expression may be the underlying cause of what we observe upon infection with E. coli or other gut manipulations. But we really still don't know how this information is being transmitted from one location to the next. And these are some of the experiments we hope to do in the future. In summary, we find that opportunistic or harmful bacteria may accelerate the development of HD phenotypes in Drosophila models of HD. These include regulation of protein clumping, mobility, and lifespan. Interestingly, these changes may be triggered in the gut and extend to the nervous system. And we're also able to regulate these changes by feeding compounds that potentially have therapeutic activity. And saying this makes the gut a potential therapeutic target for Huntington disease. Let me thank the people that have done these experiments. Angelica, Jung-Yun, Jan Ko have done most of the studies that I presented to you. Sutapa is a new postdoc that is going to take the helm and carry this project forward. I also have to acknowledge a large number of summer undergrad and research fellows and Amgen scholars that throughout the years contributed to these research using the fly model. Our collaborators are Dr. Priya Pandey, a computational biologist at City of Hope, and Dr. Larry Marsh at UC Irvine, who provided the fly models, as well as giving us advice on how to proceed with this project. Finally, and not the last, I'm very grateful to the funding sources of this project. This project was initiated by a grant from Huntington Disease Society of America and followed by gracious funding from Hereditary Disease Foundation and a discretionary fund from Caltech. Again, thank you for listening to what I babbled about. And if you have any question, I would be more than happy to answer.
So many questions.
Thank you for the opportunity.
I'm going to get started with a couple of the more basic questions. We've got 3 questions that are kind of all similar, basically about probiotics and what can we do to help our gut bacteria protect us from disease? Should we change our diet? So one of them was from Sissy Sanders, the others are from Anonymous. So it's a good question to start with.
Wonderful. First, what is a probiotic? A probiotic is practically a live organism that either in a pill form or in a solution that one could take for potential therapeutic benefits. But the unfortunate thing is that most of the probiotics out there, they have not been tested medically. So the jury is really still out whether any of the existing prebiotics have any protective value in terms of modulating our gut microbes or benefiting our organs. In terms of, what should we do? Should we take him or not? I would leave it up to you. Do your own research in terms of what that existing antibiotic is, what has been done on it. In other words, how many studies are on it, what kind of strain it is. Something that most people are not aware, for example, a lactobacillus that is found in yogurt, there are maybe over 100 different strains. And each of these strains would probably have a different property that has not been studied. So by the fact that we take it, probiotic, it doesn't necessarily mean that it has the medicinal, activities that he's supposed to have. My feelings about probiotics at this point, it's really, I don't take them, so I don't really know if they are any good ones out there until they are medically proven. But in terms of existing probiotics in nature or in a diet, for example, yogurts are a good source of probiotics that seem to be good for the gut.
Okay, there was also the question about changing the diet, and I think that's something that everybody talks about. But maybe you could comment a bit on that, Ali.
Absolutely. I presented that slide, adhering to one type of diet or another type of diet. In general, at least based on studies in humans, a high fiber diet, a diet loaded with these fermentation products seem to be beneficial for the gut microbes. On the other hand, diets that they are rich in calories and they are practically, to me, what I would call obesogenic, they are bad. So again, it is an individual, decision. Is my diet optimal for my health, or is it not optimal? I can't really answer that, but absolutely, diet, what I know is the most fundamental, tool that we have that we could change the composition and the activity of gut microbes. And in general, again, sticking to a plant-based diet rich in fibers and phytochemicals and nutrients that they have been proven to benefit us will have a dramatic effect on the composition and activity of the gut microbes.
Great. Okay. Thanks very much for that, Ali. Another question from another anonymous attendee asking, are there any plans to study probiotics or anti-inflammatory agents in human HD patients? And on that note, I actually had the question about when you were talking about putting anti-inflammatory compounds into the fruit flies, what exactly are these compounds? And is that something that humans could be taking as well, just as a precaution against future degeneration?
Let me answer your questions first. In terms of those phytochemicals that we tested here, absolutely, yes, they are present in the normal diet. And I showed a picture that these are all plant-based. And they have really wonderful properties in terms of the gut health and regulating, different cellular pathways. And one of those pathways that it is out there is really inflammation. And most of these compounds do block inflammation. In terms of the names, I thought I would leave them out because I don't want this seminar be about, advice on diet, therapeutics, or gut-based, strategies for HD patients. So yeah, I wouldn't, I'd rather not mention any names, but if someone in private would like to do so, I would be more than happy to suggest what might be good and what might not. But again, going back to, should I take this or that, it's an individual decision. One must probably contact their dietitian as well as their medical doctor and kind of analyze the diet. How is my diet now and what do I need to change? Some of us may have a perfect diet to begin with and we don't need to change anything. So in terms of adding something to existing that already good may not have some, beneficial effect. But if the diet is really, very, very way off from what it has to be to kind of support the gut health and brain health, one may need to consult a dietitian and a medical doctor to make the necessary changes. In terms of, are we going to do these products in patients? I hope we were able to do that someday. And I think these are the most immediate project we should be focusing on because it produces the data that may change the lives, within 2 to 3 years. We may not wait for a magic bullet that to have to inject into the brain over years and years to see a change. Some of these compounds may have an effect starting now. And by starting now, I mean, we don't need for FDA to approve our diet. We don't need for FDA to approve our lifestyle. We don't need, to change our behavior to benefit our health. And again, in most disorders, we're missing this gene environment interaction. While why the gene is so important, the environment is equally important. It is always a gene environment interaction. We have control over the environment, and the gut microbiome gives us that perfect system to modulate the environment to kind of reduce the toxicity of the genes or the behavior of the gene in the system. So yes, I hope that, I'm not a clinician to do these kind of studies, but I hope that, people out there that they are, able to do these kind of studies in patient, get and do these someday, yes.
Thank you, Ollie. So fascinating, and I think it's really inspiring that you're talking about things that that we, whether we are positive or whether we have HD or not, can do to control our environment, and that's really optimistic to hear, because oftentimes with HD it feels like so many things are out of one's control. So I just want to acknowledge one question, which you've already basically answered, but I did want to say a question from Yvonne Sweeten, who's saying, if you're HD positive, what can you do to have healthy bacteria? And I think you've basically just summed that up with your previous comments, and speaking to a nutritionist or a dietitian on how you can improve your diet to potentially help your gut microbiome. So we got another one here from Sarah Hernandez, who was our last presenter last month, saying, great work. I attended a talk that identified the differences in the microbiomes of patients with and without diabetes, and used a bacterial species to reduce A1C levels in the diabetic patients. Do you envision something like this that can be done for HD? Are you trying to identify a specific bacterial species that differ in HD by age of onset?
Hi, Sarah, and thank you for your question. I'm kind of familiar with that one. I think it was from Pendulum Company, that they actually have a product out there in terms of that they call, a probiotic for, diabetes patients. What I worry about is this gene environment in different groups. As I mentioned earlier, not two individuals are alike. So whether we have a bug that works in one person, it may not work in the other. And that's something really needs to be taken into consideration when you practically get one bacteria that is different between HD and a normal, non-HD individuals. What we have to also understand is, when you take a probiotic, obviously, if it's a major bacteria that is missing, that means that the niche is available for colonization. It may be a good thing to do. I have my doubts in terms of finding a specific probiotic for Huntington disease, and that is the experience that I have learned from, let's say, studies on Parkinson's disease. If you look at in Parkinson's disease, practically these variations in gut microbiomes in all over. So in terms of and that's much more advanced than Huntington disease. There are over 100 bacteria that, for example, they are different in PD patients. And these are a constellation of studies from different groups. Which one is the one that is the dominant one regulating disease will take years to accomplish. So we have to look at the gut microbiome as a holistic approach, rather than changing one, is changing the dynamics of the existing ones. Like, for example, I showed that balance at the beginning of the talk, changing the ratio of certain bacteria, like for example, good bacteria to bad bacteria, that may have more, beneficial effects than, putting one and hoping that that one will do miracle. And again, we did try that. We did put some probiotics that they were tested in Parkinson's disease models. And what was really shocking to us, it did the opposite. That probiotic was so toxic that it was even more toxic than E. coli. So it didn't have any probiotic activity in HD models. So we have to really be cautious in the context of, putting one bug or the other, or focusing one bug. And again, my, I guess, preference is always changing the dynamic of the ecosystem to benefit us. And this has to be at an early stage that, we get the benefits much faster, and that's what the beauty of gut microbe also, that it could be used as a preventative module rather than waiting that seeing the disease has occurred and then, oh, this bug is, is different in a very symptomatic HD patient. By then it might be too late. It's just not going to be, unfortunately, very effective. So if we learn to kind of modulate at an early stage, we may not need to get to the later stages that we need to put this probiotic or that probiotic.
Okay, thank you for that, Ali. And I think that's a perfect segue into two questions, one from an anonymous attendee and one from a couple from one of our grantees, Veronica Brito in Spain. The first one is, at what stage of the disease do the intestinal mutant Huntington deposits develop? And similar to that, from Veronica, congrats, interesting study. Two questions. Has it been explored in which stage of the disease that the gut dysbiosis begins? And second, you mentioned that males present more alterations than females. Can you explain that?
In terms of, the position of Huntington in the gut, we unfortunately have not done a longitudinal study to find out when do these aggregates appear in the gut. What we did simply, those slides that I showed, they are in about 6-month-old mice. They do have some symptoms, but again, we don't know if they are the byproducts of the disease, or they were initiated in the gut and later on contributing to disease. We are in the process of doing a longitudinal study to see where when they do appear, and I think that would be very, very interesting to start at an early stage and see if we could induce it by changing the gut microbiota, either inducing dysbiosis by introducing this toxic or harmful bacteria, or eliminating to see what will happen. I forgot the second question. What was the second part? Sorry.
The second question referred to males versus females, that you'd mentioned that in one of the studies, males had more alterations than the females. And if you could have any explanation for that.
Unfortunately, I cannot comment on that. That was in the studies by Julie Stout from Australia, that they found those changes, that they found that males had more variations in the gut microbes than females.
Right.
In terms of our studies,
humans.
For example, we find aggregates in both males and females, mice. In terms of our behavioral modulations, they're pretty much the same. We know it doesn't change. So we haven't seen anything in the context of male, female, at least not yet.
Right. And that study was done in humans. Is that correct? Where they found the difference
correct
between the males and the females, not in mice. Okay.
Yeah.
So I'm going to flip to a few scientific questions to make all of our scientists in the audience happy. I'm going to start with one that's not quite as scientific, but it's from Ravinder Julia, I think. Does anti-inflammatory, can it cross the blood-brain barrier? How and where do they affect the neurons in the central nervous system?
And that's again an excellent question. In fact, those that we looked at, they do not cross the gut barrier. So you see, that's very interesting, because in terms of their properties, they have been shown to change the composition of gut microbiome rather than crossing and entering in the circulations. And in fact, some of the antibiotics that we also use, they are gut specific. So meaning that they don't, cross the gut barrier into the other organs and having this side effect. So what we're thinking that these compounds might be acting is, again, by changing the dynamics of the gut microbiome. It's either by changing the ratios, and indeed we do have some evidence, even in fly, that some of these compounds change the ratio of those gut bacteria that I showed, that they have these two families of gut bacteria. So they practically change the ratio of those towards the good ones. The other thing is that we have not looked at the physiology of these gut microbes also changes in response to what you feed them. So in other words, you could tame a bad bacteria to behave good, by giving them the substrate that they may modify that bad behaviors in the bacteria and make them a bit good and less pathogenic. So I don't think the effect of these compounds that we're seeing, they have to go to the brain to see, an effect. In fact, by changing the gut, and I also maybe the reason, and I show those changes in the gene expression. We're seeing genes that they are altered in the, gut. And then we see changes that they are also altered in the brain. And the communication between these organs, the gut and brain, may be the underlying cause of the protective or the toxic effects of these manipulations that we've done.
Okay, thank you. Another scientific one from Jonathan Monkmeyer. Are the inflammatory biomarkers being studied measuring cytokine levels from integrated stress response?
We have done those in the mice, and we haven't compiled all the data in terms of what's being changed. When we looked at in the flies, we saw these clusters of genes that they are simply antibacterial. In other words, their purpose is there to ward off bacteria in a disease state. What is very puzzling to us, that regardless of how much of this antimicrobial are present, the HD flies still show dysbiosis, and they're not able to get rid of this what I call inflammatory bacteria. What you could envision that a HD flies feels that there is an infection at all time that is not able to get rid of. So in other words, it keeps producing this inflammatory and antimicrobial peptides, but yet they are ineffective. And this is what is puzzling us. We're trying to get to the bottom of this in the context of, what are these antimicrobial, peptides are doing. I gave you some analogies to a human. For example, amyloid beta in Alzheimer's disease, that's an antimicrobial peptide. Recent studies also suggest, for example, in the context of synuclein seems to have some microbial properties. So what happens to them? Then they become pathogenic. We're thinking that maybe a similar scenario occurring here, that these antimicrobial peptides, they're being produced, but they're not able to do anything, that they may end up being toxic. So this is something that we're doing, in the future.
Okay, thank you, Ali. We've got one more question. I'm conscious we have quite a few questions. Thank you, everybody in the audience who's been asking questions. I would encourage you to look up Ali directly at Caltech, Ali Khoshnan, yes, and write to him directly with your questions if they didn't get answered. But I'm going to finish up with one last question, which is very interesting and very direct, which is, where can we go to examine our gut bacteria?
There is that Human Microbiome Center, UC San Diego, with Dr. I think Rob Knights. They can even do it for you for free if you send your sample. But again and again, I will caution against this. I mean, what are we going to find out from our gut microbe? And again, if you're one, and comparing to what database are you going to make any conclusion whether your gut microbe is bad or good? But there are a lot of other industry, I think, companies that they will do that for you. But again, I'm afraid that you may not get anything out of them. So the fact that you get thousands of different species lists at different ratios one may not be able to make a conclusion whether your gut bacteria is bad or not. The question is, we don't still know what is a good bacteria or a good microbiome. What is a healthy one should look like? And a lot of studies are underway to figure out, that's what's happening. The fact that we see variation in the gut microbiome of HD patients, that may be a protective response even. We don't know if, the variation in the numbers is protective or, pathogenic. That has not been documented well to claim that. So we got a lot more studies to do to even come up with this connection that, these are the bacteria. If the bacteria are bad, is it because of, the mutation in the genes, or because of our diet, or because of something else? But again, to make the story short, and our time is running out, there are a lot of centers that you could send your gut microbe, samples to analyze your gut microbe. Yeah.
Okay, well, I know there are a lot more questions, so everyone whose questions didn't get answered, I'm sorry, but please do contact Ali or contact us and we can put you in contact with Ali. So such an interesting presentation, Ali, and very interesting to hear that there are other ways that we might be able to protect ourselves from neurodegeneration, whether or not we have HD. And really interesting that you're doing all this research about the gut microbiome and how it changes with HD. So thank you so much for taking the time to be with us today. We really appreciate it and look forward to hearing more news from your lab.
Thank you so much for the opportunity, Megan, and allowing me to spread the news about this system. It's practically an organ that we have ignored for many years, and maybe it's time to pay attention to it. It is as important as our brain, and being mindful of this organ and the ability, again, I stress that out, the ability to be able to have a certain level of control over its activity and composition is enlightening. And I think, I hope most of listeners pay attention to their gut. And at the beginning, as I said, we should really love our gut, because being able to manipulate it, it may benefit us the most. And I'm again very, very grateful to you and to HDF to sponsoring this line of research. We need more of it to be able to make better decisions about our health and the health of HD community. And again, something, I long worded here, at some point, maybe HD patients be in control of their own health. And the beauty of the gut microbiome, it could have a widespread distribution. You don't need to go to the clinic to change your gut microbiome. You could be in the most remote area of anywhere without any clinic and have a control to lessen at least the symptoms. Again, I thank you, and I hope to connect again.
Thank you very much, Ali. We really enjoyed your presentation.
Thank you so much, Megan.
So thank you everyone for attending our webinar this afternoon. Really appreciate you listening and asking all of those interesting questions. And I also would like to thank our two sponsors of this webinar, Unicure and Prilenia, both of whom are conducting clinical trials for HD right now. So thank you very much. And thank you all for attending. And please look for our next webinar next month. Thank you.